So, hello everyone, good morning. And today we are going to see a new topic that is basically derived from our previous lecture. So, in our last 2 to 3 lectures, we already studied about the carbohydrates, lipid, protein, waxes, phospholipids, glycolipids. There are several derivatives of lipids. So now this is in this lecture we are going to see how we can utilize them in our metabolic activities. So, if you wanted to see their metabolic rate activity inside our body, so you have to go through several cycles, which basically happens all the time in our body, human body, animal body, and also some of these cycles can be performed inside a plant body. So, here we have to see first in today's lecture, we are just going through the two major cycles: pentose phosphate pathway, also called as 3P cycle, glycolysis, and if we get enough time, so we can also cover the Krebs cycles. So let's start with the pentose phosphate cycle, very first and very important cycle. Actually, these cycles have a very, you can say, very important role in our plant body metabolism. Or in other words, you can say if these are not present inside our body, we are not going to ever doing our metabolic activities. So if we see the introduction part, in the case of our PPP cycle, it basically we can also call them, or you can also say that pentose phosphate cycle, also known as hexose monophosphate shunt. Okay, so we can also call them the HMP, except calling of PPP. So both the names are for the same cycle. You can call them the PPP cycle, or you can also call them the HMP cycle or HMP shunts. So now, why we are studying this particular cycle here? What is the major motive of studying this cycle? Okay, so basically, this pathway, this all this cycle, like HMP cycle. Basically, if we compare this the other cycle like glycolysis, it's more complex than the glycolysis, which we are going to study next after this PPP cycle. So why we are studying it, what it helps in. So first you can see here the formation of NADPH for synthesis of fatty acids and steroids. This is a very important key potential molecule for developing our body or synthesizing an energy, or you can say the calories. If we wanted to convert whatever the things we or eat during the day, and we wanted it to convert into the energy which we require to perform our functions, so we have to go through for this process. So that's why this NADPH formation is very needed inside our body, because if there will be no formation of NADPH will take place, so hence there will be no formation of fatty acid will take place, or if there will be no formation of fatty acid take place, then there will be no formation of beta oxidation in mitochondria, and hence they have to produce ATPs. So that's why these all processes are very connected. You can't even feel the body are going through lots of cycles and pathways during they are taking just breathe in and breathe out. So basically this is the first one. And second is the maintaining reduced glutathione for antioxidant activity. So this glutathione, this is also a very important molecule. So basically, when this cycle happens, this hexose monophosphate cycle happens, and relatively other cycles happening in our body, what they do, they get oxidized this glutathione inside our body. So reduction and oxidation of this glutathione can be dangerous for us activity. So basically, their reduction, the reduced glutathione inside a cell, also you can say that these are HMP process. This is also responsible for maintaining the quantity of this glutathione for the antioxidant activities. So that's why this is also very important. Now we have to see the third thing, which is a very good byproduct we are getting from our PPP cycle. Synthesis of ribose for nucleotide and nucleic acid formation. So, DNA is a basic core unit of our every activity because DNA is a coding unit, it's the information unit. Anything, any function which is performing inside our body that is basically governed by the DNA activity. So, this ribose sugar, this is the main component of forming, or you can say, the formation of nucleotide or nucleic acid. So, if there will be no ribose, hence there will be a very complex situation will be formed for the formation of nucleotide and nucleic acid. So these are our major motives, like why we are studying this PPB cycle. Hence, you know that this is very important cycle for regulation, for normal regulation of our body activities. So that's why these are the three fundamental things on the basis why we are studying it. Now, the learning objectives, we already know the importance. So, hence, here our learning objectives will be to study the meaning type of pentose phosphate pathways and glycolysis cycle. And secondly, we have to see their specific steps. Okay, a pathway has several steps to follow to finding the last product. So we have to see every step very carefully because if you are going to miss a single step in between, you are very complicated. It will be to find the last one. So we have to be very careful. It's like a mathematics. You cannot skip a step. Okay, you cannot overtake that. You have to be very careful about each and every step, what kind of enzyme, intermediate activities, what is reduction, what is gaining. So every step you have to be very careful when you're going to study these steps. You have to be very mentate for that. Last, in our learning objectives, to understand importance and role in human body. That's our major concern. That's why we are studying all these, right? So this is very important that we have to know why we are studying it. What are the major purpose of these pathways inside our body? So let's start with the phosphate pentose phosphate pathway. First of all, if we see it here, so basically, as of our process glycolysis, we'll study it later. So glycolysis happen inside a cytosol in the cell. Same, the pentose phosphate pathway. This is also happen inside our cytosol. So in the cell, of course, the cytosolic activity is also governed here. So oxidation is achieved by dehydrogenation using NADP, not NAD. This is very key point. You have to remember it. Here, oxidation will be achieved whenever the cell, whenever any molecule getting oxidized here. This is by dehydrogenation or using NADP, not NAD. Okay, in many cycles like even glycolysis and other cycles, you are going to use the NAD. So here you have to be very careful. We are not going to use NAD. We have to use the NADP. So in this process, there are two major steps. Means when you are going to study the pentose phosphate pathways, there are the two major steps which have to be followed. So the first step here, irreversible oxidative phase. First step is irreversible oxidative phase means whatever the reaction which is going under this oxidative phase cannot be reversible. Okay, once a product can be formed, it cannot reverse in their original form. So whatever the thing has been changed, it will remain changed. So this is the first thing. Second step here is a reversible non-oxidative phase. So that is quite opposite to the first one. Firstly, it is non-oxidative. They do not require oxygen molecule here. And secondly, they can be reversible. So whatever the product which is formed inside the reversible, or you can say whatever the form product will be delivered after every step, it can reverse back to their original form. So in this, rearranged of regenerate two molecules of glucose 6 phosphate and one molecule of glyceraldehyde 3 phosphate. So look, it may be sometimes you feel that this is very complex, like having oxidation and reduction. Again, they can be reversible. Sometimes they cannot be reversible. So, whatever the main purpose of this pentose phosphate sugar which they are doing here is they're just playing the carbon numbers. Okay, they have to perform the carbon detail about, like in the first oxidative phase, they just what need to do, they have to secure a position from carbon 6 number to carbon 5, as they all are the organic one, so that they are having the carbon inside. So what their basic motto here, they just degrade their carbon numbers, and for degradation of their carbon number they have to perform these following steps. Okay, so these all the things which they are doing here, their major motto is here to do that for the reduction of that carbon particular atom from the sixth position to 5. And in the reversible non-oxidative phase, they can also do it like they can be the from the sixth number to even third carbon number. Okay, 6 to 5, 5 to 7, 7 to third. So these they can follow up these details just to adjust their carbon number according to their need. So basically, whenever the thing is required inside a cell, that basically their functionality is based on their structure. So hence, if they want a different kind of function, so hence they need a different kind of structure. So that's why this structure metabolism can change according to the demand. And basically, this reversible non-oxidative phase in pentose phosphate sugar is totally demandable. If cell demand some several kind of function inside the cell, only then they perform the function. They do not unnecessarily provide anything when the cell requires. They only work when the cell asks for it. Okay, if they are getting a signal molecule to perform such kind of function, then only they do undergo the phase of non-oxidative phase. So this don't get confused. Okay, just be very clear about both the phases. Now we will see the first oxidative phase here. So in the oxidative first phase, okay, you can see here there are just two things, or you can say that there are just two major reaction you can see here. First, our thing which we start the function is glucose 6 phosphate. Okay, so glucose have a 6 carbon number, so that's why this is glucose 6 phosphate molecule. Now what we need here, we need here is the NADP, or now the process that here happens is basically dehydrogenation of glucose 6 phosphate. So when we do the dehydrogenation, you can see here the water molecule is added here. So whenever the dehydrogenation of this glucose 6 phosphate will be converted into 6 phosphogluconate. Okay, when the dehydrogenation of glucose 6 phosphate happens, what we are getting here? We are getting here 6 phosphogluconate, and in this reaction, this is a reaction, this is a metabolic reaction. So we need an enzyme to basically motivate this reaction or catalyze this reaction to happen and obtain their final product. So this catalyzed by the glucose 6 phosphate dehydrogenase. You have to be remember these enzymes. Okay, so whatever the thing and what that converted into and what are the byproduct they are getting at that particular reaction, and what is the enzyme which basically catalyzes that reaction. So these are the points you have to remember. Okay, don't forget to remember these enzymes. This is very important. So glucose 6 phosphodehydrogenase, this is the first enzyme which use this glucose 6 phosphate to become 6 phosphogluconate. In this reaction, you can see one NADP will convert it into NADPH by the dehydrogenation process. Like you can see easily see here, this H is converted here. Yes. So the NADP will be known as the NADPH because they just add around one hydrogen atom here. Simple. Next, followed by hydrolysis of 6 phosphoglucolactone to the, you can see here next, and the final thing we are getting in this cycle is ribulose 5 phosphate. Okay, 6 phosphogluconate is then catalyzed by the 6 phosphogluconodehydrogenase, and that will be converted into ribulose 5 phosphate. Okay, now you can see here the carbon number is changing. Okay, carbon number 6 here, then this hydrogenase, okay, this gluconate dehydrogenase, the enzyme which used here, that basically mandate that particular 6 phosphogluconate to reduce a carbon dioxide from it. If they are reducing a carbon dioxide as a byproduct with along with NADP to NADPH, and simultaneously a carbon dioxide atom will be released from here. So what we are getting here? We are getting here from the 6 carbon to the 5 carbon. And this is our first, you can say this is our first success. Like we are getting a ribose 5 phosphate, that is a carbon 5 thing, and we needed it to formation of nucleic acid and several other parameters. So this is a major motive. You have to remember this, okay? Here you cannot forget that. That is a very important detail. Next is a non-oxidative phase here. So ribulase 5-phosphate then will be converted. We will take it by, I think, diagram will be more appropriate, because this is a theory you can get confused. Right. Let me just enhance it a little that you can easily visible. Okay. So till here we get ribulase 5-phosphate in the oxidative phase. Now the non-oxidative reversible phase has been started here. So in this case, you have to remember now the carbon number do not get degrade. Okay, only one carbon number degrades in the oxidative phase, but in the phase of non-oxidative reversible phase, whatever the carbon number has initiated in the first form, like you can see, now there are two divergent. Okay, one is this ribulase 5-phosphatase 3 epoenzyme. This epienzyme used here first for this production of xylulose 5-phosphate. There will be two enzyme work on ribulase enzyme. Okay, one enzyme for formation of the xylulose 5-phosphate, second ribolinase enzyme for making the ribose 5-phosphate. The same thing, which you can say just only they are one enzyme activity here, that's why this ribulase is converted into ribose 5-phosphate, and that we need for the nucleic acid formation. So this ribulase phosphate, now you can see here whatever the further step are taking place here, but still the carbon number is always same. Like you can see here, xylulose 5-phosphate and ribose 5-phosphate. Total carbon number is 10. Next, they are converting it to pseudophylloxase 7-phosphate or glyceryl aldehyde 3-phosphate. Even though their carbon numbers are same, 7 and 3. You can see here. Next phase, they will convert it into the erythrosase 4-phosphate plus fructose 6-phosphate. Same, the carbon number is 10, 4 to 6. Now they can make the fructose 6-phosphate and fructose 6-phosphate. Still, the carbon number can be the here the carbon number can be enhanced because this is the product we want. After that, if thing can be converted into glucose 6-phosphate, then we can use that particular things, because our motive was for that. We have to convert that glucose 6-phosphate to the fructose 6-phosphate. So converting this thing, we cannot lose our one carbon. So basically, if this reaction getting engaged in last with the glyceraldehyde 3-phosphate, dehydrogenase acetone phosphate, then only they are getting the 6 carbon number in a final product. So this is all about our pentose phosphate pathway. Basically, what you have to remember, the key thing is oxidative and non-oxidative things, and the major why we are doing this, because we need NADPH for the formation of fatty acids and other steroids, and also we are getting here the ribose 5-phosphate, which is a major component of the nucleic acid for formation of further genetic activities. So these are the basic motto here, that why we are studying this pentose pathway. So now we will see the some you can say their activities, like why we are important and importance of pentose for sweet sugar. A little theoretical version of these all things. So importance of NADPH. Like we studied this and we got NADPH in our pathway. So firstly, what is the function of NADPH here? So as we got a NADPH, so first function is to present the biosynthesis of fatty acids. So fatty acid, as I already told you in our introduction part, like fatty acids is very important because the beta oxidation of fatty acid inside the mitochondria produce ATP. And all we need for doing functions is ATP. That's why this is very important thing. Secondly, certain amino acids involve the enzyme glutamate dehydrogenase, which we get during this NADPH formation. So that basically this enzyme is involved in many, you can say many pathways for relying their reaction. Thirdly, antioxidant reactions. So I already told you in the introduction part also about the glutathione. So basically that mediated reaction of water. So water have to be do not reduce the glutathione in the cell membrane, because reduction of this glutathione may be cause several harmful impact on the cell membrane. Detoxification of drugs like cytochrome P450, P440. So detoxification of drug is can be done by NADPH. So this is very important. We have to detoxify drugs. Phagocytosis. Phagocytosis is also a very important term. Like sometimes the cell can eat up itself. So at that time, we need to stop doing that because it can let the dead of the cell. So that's why at that time also NADPH help them to release the energy and make up them capable of working again. Integrity of RBC membrane. So the cell, RBC cell, basically when NADPH release, they are very helpful for maintaining the balance of the cell membrane. Like what amount of the things can be taken inside and give outside. So these are the following things which is very important in the case of when we talk about the NADPH importance. Next here, importance of pentose sugar. As we all know, ribose 5-phosphate, very useful for the synthesis of nucleic acid, RNA and DNA. These are very important things for our body. Skeletal muscles capable of synthesizing pentose. So these skeletal muscles, okay, everything we do, that requirement of the muscles have to be there. So that's why these pentose formation is also a part of these particular skeletal muscles. So skeletal muscle, you can see when synthesizing the pentose, that will helpful in a very way more activities of the metabolic pathway in our body. Next, we will see here clinical aspects. So, clinical aspects is also related in our human body because, in disease, or you can say if the pathway has some problem, okay, like if we are facing some problem like genetic problem, genetics defect of glucose 6 phosphate dehydrogenase, implement of the generation of NADPH X chromosomes. So basically, if they are having any problem in the NADPH synthesis and there is no ribose sugar will be take place, so the vice versa effect will be can come into our genetics problem. So if we wanted to control our gene and work in a very efficient manner, so we really have to work upon it. Mediterranean and Afro-Cambrican origin, red cell homolysis subjected to oxidative stress. So these are the basic things which is very important, but this all thing can be go smoothly only if the NADPH regulation in our body is in accurate manner. Or how the NADPH will come in accurate when we have a regulatory and normal pentose pathway in our body. So, subject to oxidative stress in the infection, drugs such as anti-malarial and sulfamides, favabeans, favism. So these are the basic, you can say, the disease which can be happen inside our body if something gets wrong. So these are some references from taking these material, and you can also go through these references as I will upload your PPT on dialysis, and you can go through this. These are really very good papers and chapters from a book, and take a look. This will be helpful in your further studies of biochemistry also, like this Harper's Illustrated Biochemistry, a very good book and very deep knowledge about how you can, step by step, you can study every cycle, whatever you wanted to study, and their principles and how they are very useful in our body. So these things you can be go through, and this will be good. Next cycle here. If you have any problem regarding the pentose phosphate pathway, PP pathway or HMP pathway, so you can text me or message me, or you can even ask me after the lecture, like, ma'am, this thing not get understandable, so I can explain you later. So glycolysis, very interesting. It's like important, interesting subject or interesting cycle basically. So if we are talking about the glycolysis, it's all started from the glucose, a six carbon sugar. So all the things, or you can say glycolysis is basically the definition written here is well understandable. Like glycolysis is a stepwise degradation of glucose, okay, and other simple sugar, not only glucose, but all other simple sugar also can be degraded by our process of glycolysis. So same as like PEP process or pentose phosphate pathway, this process is also happen in the cytosol of the cell. It is unique, and in that it can function either aerobically and anaerobically. Why it is unique? Because it can function in the presence of oxygen, and it can also function in the absence of oxygen. This is rare. Not every cycle can go through like that. They can either require oxygen or not. Not the one single cycle can go through both the phases, but this glycolysis, you can see here, they can go both the ways. Either if the oxygen present, good. If not present, then even more good. They can go through with both the processes. Now, if we talk about glycolysis, like their ontogeny, where they come from, so this is glycolys means sweet, and it's a Greek word basically, and lysis means splitting. So basically, a sweet thing which we are going to split here. Next, we will see how we can split it here in two phases, basically. If we talk about the glycolysis, like it's okay that we are going step by step, but basically there are 10 reactions. 10 reaction, 10 intermediate reactions, and the final product of glycolysis is pyruvate. What we want here, like we are breaking down the glucose, or we are breaking down the sugar molecule in the simple version, but our demanding thing, what we want as a byproduct in a glycolysis, this is the pyruvate. Or for obtaining this pyruvate or getting this pyruvate, we have to go through 10 different reactions and 10 intermediate reactions. This is huge but simple. This is broad one, but not as complex, because complexity is basically, if we compare it to the pentose phosphate sugar, then numbers are more, but the complexity is less. So here you can see here in the first a series of five reaction, glucose is break down in two molecules. So a total five reaction is happened here for just to do the glyceraldehyde 3 phosphate formation. Okay, so just the glucose is break down into two molecule of glyceraldehyde 3 phosphate. This is the desirable product in the starting five reactions. In second phase, five subsequent reaction convert these two molecules, the one which we are getting from the first five reaction, glyceraldehyde 3-phosphate into 2 molecule of pyruvate. That is a thing which we wanted. So we have to go through these 10 phases. First 5 phases for obtaining glyceraldehyde 3-phosphate 2 molecules. Second 5 phases by the subsequent these reaction for the glyceraldehyde 3-phosphate into the 2 molecule of pyruvate. Now you can see here, phase 1 consume 2 molecules of ATP. Okay, now we have to see like how much ATP can be consumed and how much ATP can be produced. Both the things happen in this cycle. ATP can be produced, ATP can be consumed. So mostly there is a 10 total number, I think. Yeah, 10 total number of ATP can be produced inside the this glycolysis pathway, but 2 will be consumed. So there will be only 8 ATP you can get in here in a production version, final product. So the later stages of glycolysis result in the production of 4 molecule of ATP. Okay, and the net is 4 is equal to 2 is equal to 2 molecule of ATP produced per molecule of glucose. So you can see here there will be the 8 molecules which will we are getting in a glycolysis pathway. So now we will see the first phase of the glycolysis, or you can say the reaction 1. So reaction 1, phosphorylation of glucose by hexokinase or glucokinase. So there are two enzymes which are going to help us to convert the glucose into next upcoming product. So the glucose enter glycolysis by phosphorylation of glucose 6-phosphate catalyzed by hexokinase using ATP as the phosphate donor. So, and under this physiological condition, the phosphorylation of glucose to glucose 6-phosphate can be regarded as irreversible. So this very first reaction of glycolysis, in which what we are doing the phosphorylation of glucose 6-phosphate. Okay, so glucose to the glucose 6-phosphate, and here we need a ATP basically as for the phosphate donor, because we need as we are having glucose which has a 6 carbon number, but what we have to do, we have to add a phosphate group inside here. So for adding that phosphate group we need a ATP, and that basic ATP is giving us phosphate to add on in the second reaction. Let's see what are the significance of this primary reaction. I think we can take it later, the significance part. Maybe we will take the cycle actually. We're going to, yeah, here it is. So these are the 10 reactions I was talking about when we are talking about the glycolysis. Okay, so you can see here it is started from this pink version, the glucose. Okay, so you can easily see here first a very important is glucose. Okay, we have to convert this glucose into glucose 6 phosphate. In short, we can say them G6P. So this G6P, basically for the phosphorylation of this thing, we need a ATP. You can see in a very first reaction, this is the ATP here, and that converted into ADP. ATP means adenosine triphosphate. There are the 3 molecules of phosphate present in an ATP. But now, as it gives is one phosphate for making the glucose to the phosphatizing agent, that's why there's just left with the one ADP. So in this reaction, our enzyme which help us to conversion, that is hexokinase. Okay. This is the first reaction. Now we will see the second reaction here, from the glucose 6 phosphate to the fructose 6 phosphate. So in this reaction, we need to change the configuration from glucose to the fructose 6 phosphate. And what enzyme which is helping us to make this kind of product is phosphoglucose isomerase. Okay, so if now the enzyme is isomerase, which is helping us to the conversion of the glucose 6 phosphate to the fructose 6 phosphate. Now In this reaction, we are not neither gaining the ATP, neither removal of the ATP happens here. Now the one, third reaction here, where we have to convert what the product glucose 6-phosphate to the fructose 1,6-bis biphosphate. For this reaction, in this reaction, we will convert it in the ATP to the ATP. Now, in the whole four reactions, which we already studying here, we can see we lose or we consume the two ATPs. First, in the very first reaction, glucose when converting into the glucose 6-phosphate, and second, when the fructose 6-phosphate is converting into the fructose 1,6-biphosphate. So you can see this type of reduction division happens here. Okay. So in this reaction, what the enzyme which helping us is a phosphofructose kinase. Okay. The phosphofructose kinase is helping us to conversion into 1,6-biphosphate. Now, from this fructose 1,6-biphosphate, now the most exergenic reaction, these four reactions which you studied, these are the endogenic reaction. Now the exogenic reaction happens. Mostly why we call these the endogenic reaction, because they are consuming ATP. That's why we are calling them the androgenic reaction. But now here you can see they are exogenic reaction because now in this case they are producing ATPs, or you can say they are producing the energy. So after this four, aldose is starting their work, and what they are doing here, they are converting to the fructose 1,6 biphosphate to the triose phosphate isomerase, through triose phosphate isomerase to the glyceraldehyde 3 phosphate dehydrogenase. I know names are quite tough, but it's okay. From this, we are getting 1,3 biphosphoglycerate. It is a BPG. This is very, you can say this is like a backbone of this whole glycolysis process, because after this process we are getting two ATPs, because the first in that there we are getting the glyceraldehyde 3 phosphatase after the aldose reaction, and that time you can say this is a reversible reaction here. It can be dehydrogenate acetone phosphate DAP at G3. Both the things can be performed on the basis of triose phosphate isomerase enzyme functionality, but it can be reversible. So after this reversible thing, the enzyme here, like the glyceraldehyde 3 phosphate dehydrogenase, basically helping us to converting them into the 1,3 bis phosphoglycerate BPG. So in this reaction, for occurring this BPG product, we need to convert the NAD to the NADH. Okay, at that time, for happening that, they have to lose one hydrogen atom and converting NAD to the NADH. So when they losing that particular hydrogen thing there, so that's why the number of, like you can see the number of the bisphosphate is like 1,3. Like one particular hydrogen atom is added here. That's why this 1,3 bisphosphoglycerate BPG formed. After this 1,3 by bisphosphoglycerate have been formed here. Now there is one another enzyme showed their activity, phosphoglycerate kinase, which basically help this BPG to convert into 3PG, or you can say 3-phosphoglycerate. Here the biphospho, okay, these biphospho it basically removed, like ADP convert into the ATP, because one phosphate thing, you can say a biphosphate means they are having the two molecule of phosphate inside them. So one phosphate will be going to add on to the ADP, and ADP addition will be led to the beginning of the ATP formation, because now they are having the 3-phosphate in the ADP. So this is the first time we are getting the ATP here, or hence we are getting a two molecules of ATP here. All right, so it's the two molecules. Now 3-phosphoglycerate PG. Then again, enzyme mutase help them to convert it into 3 to 2 PG reaction. Okay, So 2-PG is 2-phosphoglycerate. Now 2-phosphoglycerate will hence by the activity of anolase, they will be converted into phosphophenol pyruvate. This is almost when we are going to have our final product pyruvate. So at that time, these phosphophenol pyruvate, for happening that, we need a final enzyme here. The enzyme to get a final product, pyruvate kinase. So this pyruvate kinase, what they do here, when their enzymatic activity is starting up, so phosphophenol pyruvate, okay. So basically, this 2-phosphate will be go to the ADP and converted to the two ADPs. So now this phosphate group is reduced and pyruvate kinase take their activity. That's why that phosphophenol pyruvate will be converted into 2-pyruvate molecule, because phosphate group has been reduced from there, only pyruvate group has been remained there. So hence, as a product, we are getting in the glycolysis is 2-pyruvate, and this is our main motive. Get here is glycolysis as a pyruvate, because pyruvate is a channeling molecule for many activities. We will see there what in what things the pyruvate can be very helpful. Now, a little detailed version you can see here. This is all the theoretical thing is written in this slides. What I already told you, like first timing reaction. This thing will additional submolecules sometime, like the magnesium ions is very important in handling these activities. Hexokinase, or you can say the first priming reaction of the glycolysis. At that time, some cofactors also work to enhance the reaction. At that time, magnesium ions play a very important role for their activity. This glucokinesis, the thing which is the first product or the first reaction in the glycolysis, that occurs in the cell of liver, pancreas, gut, and the brain of human and most other vertebrates. So this is very common thing. I think you should know till now that these things have to be present. So the fate of glucose 6-phosphate, the first reaction fate, what you studied, so they can be helpful in these all pathways which have been written here. Like they're very useful in glycolysis because we have to move forward with this reaction. Gluconeogenesis, the glycogen deactivation, pentose phosphate pathway. This is very important because the activity of our pentose phosphate pathway has been initiated with the glucose 6-phosphate. Glycogenesis, glyconeogenesis, glycogenolysis. These are the processes that are basically based for the sugar reduction process in our body. Then at all these reactions, glucose 6-phosphate plays a very important role for initiating, intermediating, or finalizing their results. So you can say this is an important one. Now this is a yield, like what ATP per molecules we are getting in the glucose. So first we get the 6 molecules, pento phosphodiesterase kinase we are getting 2, pyruvate kinase we are getting 2, to total 10 number of molecule of ATP we are getting in this whole reaction. Okay, but in a starting, in a very first reaction, in 4 reactions, we already reduced to ATP molecules. Hence the net ATP yield in the whole glycogenesis process is 8. I think you is clear. Now the significance of glycolysis, why we are doing this. It's all about like energy production. It's okay, but now we have to see the what are the some other significance of glycolysis, even other than the energy production. So you can see here, glucose 6-phosphate is a very fundamental first thing which we are getting from this reaction, glycolysis pathway. That is intermediate of member of pathways like we already studied, the HMP pathway, glycogen analysis. So this is very important for also synthesis of glucoamines. So glucosamines synthesis can be done only with the help of fructose 6-phosphate. So that fructose 6-phosphate is a product of we can have it by the glycolysis. So this is very important. Triose like glyceraldehyde, 3-phosphate, and other glycolytic intermediate can be used in HMP pathways for the production of pentose. So when we studied the pentose phosphate pathway, at that time we need the glyceraldehyde, or you can say that we need the pentose there for their production activity. So this thing also helpful in that pentose pathway. So there are several pathways in which the product or the byproduct or the intermediate product of glycolysis can help for activating their roles. There are also some three fates of pyruvate produced by glycolysis. So whatever the final product, pyruvate of glycolysis, they can be function in the anaerobic condition and do the lactic acid formation. Lactic acid formation in anaerobic condition can be done by pyruvate. In aerobic condition, they can do the citric cycle, and in the anaerobic condition, they can do the alcohol fermentation. These are the very useful things which can be done by the pyruvate, which is the finalizing product of glycolysis. So now this is some questions related to the pentose pathways and glycolysis both. And I think if you're going to understand the unit or the cycle, so you can easily be answerable for these questions. I will upload these questions on your LMS, and whenever you get time, take a look. And if you are not getting answers, so please just tell me. Like I will explain it if something left over. So basically, these are the very relevant one which you studied, and if you can answer these questions, so it's like the 70 to 80% you are getting it correctly. This is the reference for the glycolysis, as I already give the reference for the phosphate pentose pathway earlier in between when we ended the topic. So you can see here, these are the some very good biochemistry books on which if you go through, then you will find several other cycles and pathways and metabolic activity reference. So this will be very helpful for you to understand our body metabolic activities. So this is all about pathways and cycles, and I hope you understand it all. But still, if you guys any problem, just let me know. I will try to help you out with your problems. Okay.